Good afternoon, everyone. Thank you for joining this session. And actually, thank you for choosing this session, since it's a people choice session on ADCs, antibody-drug conjugates. I'm Paolo Tarantino from Dana-Farber Cancer Institute, and I would like to thank, beyond you, also the organizers for for allowing this session to happen, because I think there's a lot to say about ADCs, about their past, their current utilization, and their future. And we have an outstanding panel of experts that are going to guide us through every phase of ADCs development. And so, without further ado, I would like to invite Professor John Lambert, PhD, one of the legends that has been working on ADCs since the early 80s. He has been CSO of Immunogen, has led development of TDM1 and mirvetuximab, among other ADCs, and he's still working in the ADC field. And he's going to go through how ADCs work, how ADCs behave in the body, what is their history, where they come from, which is going to help to understand how to use them today and how their future will look like. Without further ado, please, Dr. Lambert.
Thank you, Paolo, and I thank the organizers for the honor of speaking here. So these are my disclosures. So the aim of an ADC is to increase the therapeutic window for oncology drugs. And as you all know, the cytotoxic chemotherapy essentially has the maximum tolerated doses and the effective doses are the same. And the hope was to increase the maximum tolerated dose by decreasing systemic toxicity via altered distribution and by decreasing the minimum effective dose via specific delivery to tumor via antibody binding. Now, a biologist's view of an ADC is that conjugation of drugs confers potency to an anti-cancer antibody that might otherwise not be active enough. But the biologist's view, one has to also bear in mind that biologists they only think about the effect of antibody binding to the target, and they don't never ever worry about catabolism. And catabolites because the catabolites are amino acids. Now, a medicinal chemist view of an ADC is that it's a type of prodrug. Here's a drug, and here's the antibody, which, as a hydrophilic moiety, prevents the transport of the hydrophobic drug across cell membranes. It could be a phosphate group. However, medicinal chemists need to think about the constraints of delivery conferred by antigen density and also the altered distribution of catabolism via conjugation of the antibody. So the basic components of an ADC, you have the antibody, you have the drug, the payload. Well, you obviously need a method of attachment, conjugation. Sometimes that might be all you need, but usually there's a spacer to actually push the drug further away from the protein molecule. There can be a trigger release mechanism or not. And there can be a self-immolative release moiety or not, so that the drug could be released in unchanged form or with a bit of the linker still attached. And this is how they work. The ADC will bind to a tumor-associated antigen, be internalized into an endosome. Now, what happens next? Actually, it can recycle back to the plasma space again via FcRn binding. Some targets recycle quite readily, but any that ends up in the lysosome, the antibody and linker gets degraded, releasing the drug, so the drug can exert its effect. And if the payload is hydrophobic and can cross membranes, it can actually exert effects on neighboring cells as well via a bystander effect. So some of the history and early key lessons learned. Well, ADCs have a very long history. Even in the late 1880s, Paul Ehrlich envisaged a magic bullet whereby a toxic agent could actually have as part of it an agent of specificity. And in the 1960s, when chemotherapy was available, people conjugated chemotherapeutic agents to IgG fractions. But it was really the invention of monoclonal antibodies that provided the compounds that had the selective binding envisaged by Ehrlich. And of course, this invention earned Milstein and Kohler, along with Niels Jern, the Nobel Prize for Physiology or Medicine. So, as I say, they have a long history. Immunogen and my part of this history started in 1981 when Immunogen was founded, and I was the second scientist hired on the project. And Emil Frey, whose picture has already appeared at this meeting, was one of the scientific advisory board members, and he said the goal is to improve the efficacy of anti-cancer drugs just by a factor of 2 or 3, and you could have a great drug. And so, hence ADCs. So, what were the key lessons learned? First, in the 1980s, dosimetry studies with radio-labeled antibodies, people could measure how much ended up in the tumor. And actually, it doesn't matter, across lots of tumors, lots of antigens, many different antibodies, about 0.01% of the injected dose per gram of tumor accumulates in the tumor 1 day after IV administration. And that's approximately the peak amount that ends up in the tumor. So if you had a kilo of tumor, it's only going to take up 10% of your drug. But the other consequence of that is that the first generation of ADCs in the 1980s used standard anti-cancer drugs. It was often thought that conjugation of cancer drugs to antibodies could be a line extension to increase efficacy. But it turned out when you do the math of how much antibody actually ends up in tumor tissue, it was clear that you need highly potent payloads. The third key lesson was that the first generation of ADCs used murine monoclonal antibodies. Even the immunologists in the very early 1980s didn't think that mouse antibodies would be immunogenic, but they were. Now, the immunogenicity thing was solved in the late 80s. For example, this paper, Reshaping Human Antibodies for Therapy, there were then new ways of making fully human antibodies using phage display. And for this work, Greg Winter earned a Nobel Prize, sharing it with 2 others. So now the first ADCs to receive approval. Well, first one has to think of how long it takes. From the invention of monoclonal antibody therapy for the first publication, it took more than 20 years before the first antibody drug was approved in a cancer indication. That was rituximab. Shortly thereafter, the first ADC was approved for the treatment of AML. It was an anti-CD33 humanized antibody that binds to a myeloid target, conjugated to this really, really toxic DNA-binding drug that binds the DNA and causes DNA double-stranded breaks. It was approved in the year 2000. And the approved dose was 9 milligram per meter squared, or 0.24 mg per kg, for 2 doses, 2 weeks apart, and that was it. The drug was so toxic you couldn't give more, but it was enough to have provide some clinical benefit to AML cells. But Immunogen started to look at potent tubulin-acting agents, maytansine as an ADC payload. So when the R group is hydrogen, that's the parent drug maytansine, and Ravi Chari, the chemist in the group, developed adding 1 extra carbon and a thiol, or 4 extra carbons and a thiol for DM1 or DM4. And the thiol group, the sulfhydryl group, provided the chemical handle to be able to conjugate to antibodies. And so this work was published and developed and published during the 90s. And in December of 1999, the very first patient dosed with an antibody containing one of these potent tubulin binding agents was dosed here in San Antonio by Dr. Tolscher. It was an antibody that bound to a target on colon cancer, which actually ultimately led to another important lesson for ADCs: match the payload with the disease. So tubulin agents were not active in colorectal cancer, and it wasn't for lack of trying. And so if a class of cytotoxic agents is not active in a particular disease, one shouldn't expect antibody-mediated delivery to change this. Now, Seattle Genetics, which was founded towards the end of the 90s, also developed a potent tubulin-acting agent based on auristatin E as the ADC payload. To create the chemical handle, they knocked off one of the methyl groups from the tertiary amine here to make a secondary amine, and that provided the chemical handle for attachment to antibodies. And this was developed by the chemist Peter Center. And brentuximab vedotin, brentuximab is an anti-CD30 antibody. It entered the clinic in 2006, again in Texas at the MD Anderson Cancer Center with Dr. Eunice as the PI. So I'll just show you one example of the type of data that we and at Immunogen and Seagen were developing with these potent tubulin agents. This is trastuzumab, which Genentech provided, linked to DM1, the maytansinoid, via an uncleavable link through lysine conjugation. And this is the type of data that got Genentech excited to go forward with taking this molecule to the clinic. So this is a mouse xenograft model, but with human HER2, and it's sensitive to trastuzumab. This is weekly doses of 15 mg per kg. As soon as you stop dosing, the tumor takes off again. It's very aggressive. But a single dose of 15 mg per kg TDM1 eradicated the tumor. Now, I always like to think that 2010 really marked the turning point of ADC technology. So after 10 years on the market, Pfizer withdrew Mylotarg, as its brand name was, gemtuzumab ozogamycin from the market after a failed phase 3. And in truth, this ADC was really very toxic. It didn't have the properties that would lend itself to being able to treat solid tumors. And that was in June of 2010. But also in June 2010, the phase 1 study of TDM1 was published. And the major findings was that the DLT was transient grade 3 thrombocytopenia. The recommended phase 2 dose, 3.6 mg per kg, once every 3 weeks. And in this dose-escalating phase 1 trial, there were 5 confirmed partial responses. And also in November, also in 2010, but in November, brentuximab vedotin phase 1 was published in New England Journal of Medicine. The DLT of this was reversible neutropenia. The overall response rate was 38% in this dose escalation study. And these phase 1 data ultimately led to approvals. So TDM1 was the first ADC to receive full approval from FDA based on a randomized phase 3 in 2013. And brentuximab vedotin received accelerated approval for treating Hodgkin's lymphoma and acute large cell lymphoma in 2011, and received full approval based on a randomized phase 3 in 2015. And in the early then 2010s, data was emerging that these potent tubulin agents really did have the properties that everyone in the ADC field sought after, that is, good activity with well-tolerated doses. And this sparked a whole bunch of innovation. And a decade later, the fruits of that are these 8 approvals in the last 5 years, the most recent one being mirvetuximab soravtansine from Immunogen. Also, a note here that Mylotarg got reapproved on a fractionated dosing schedule that allowed for a lower toxicity. Now, this shows the explosive growth. The blue lines represent new ADCs entering into the clinic for the first time. And you can see that after the early data of 2010, during the 2010s, around 20 molecules entered the clinic per year, new molecules. In 2019-20, suddenly this took off again until this year in November, which is when this slide was put together, there were already 70 new ADCs entered into clinical trials. The other thing to note here is that 5 years ago, most of the ADCs that entered clinical trials used potent tubulin agents, auristatins or maytansinoids. And there was just one with a topoisomerase inhibitor. And look what happened in 5 years. By now, 3/4 of the ADCs that entered into clinic actually used potent topoisomerase inhibitors versus the potent tubulin agents. Now, why all this interest in topo-1s? Well, I don't need to really tell this audience why there's a great interest. It's the development of trastuzumab deruxtecan, which is a topoisomerase inhibitor exatecan linked via a tetrapeptide and a short spacer. And when the tetrapeptide gets cleaved, some of the spacer falls off, but it leaves a derivative of exatecan called DXD. And this is the type of data. This was in New England Journal of Medicine 2022, TDXD against TDM1, which is already a good drug for HER2-positive breast cancer. So what's next? The ADC development today and tomorrow. So the goal really is to further improve the toxicity profile of ADCs and to increase the therapeutic index and perhaps begin to address mechanisms of resistance. So it's important to come back again to ADC catabolic pathways. So when you inject an ADC into the bloodstream, some of it will end up in the tumor, but remember, even if I had a kilo of tumor, it's probably at best going to take up 10% of the ADC. The other 90% is going to be catabolized somewhere else in the body by normal tissue. Now, in the first generation ADCs, where the linkers were perhaps unstable, the conjugation sites were certainly unstable, and some of the payloads were quite hydrophobic, so that accelerated uptake and catabolism. It's not surprising you would have payload toxicity. So think of the 4 approved vedotin ADCs. The tox profiles are neutropenia and neuropathy, and that's the toxicity profile of dolastatin 10. Now, I would say that in new generation ADCs, this is largely a solved problem. You can have perfectly stable conjugation, stable linkers, no hydrophobicity. And that actually leaves these two mechanisms and the elimination pathways as sites of toxicity, especially the non-target uptake by fluid phase pinocytosis, which is the normal antibody catabolism. It's taken up by all sorts of cells and at a certain low rate catabolized. I won't really address on-target toxicity, the unwanted on-target effects. But if you think of the potent tubulin agents, when they're linked in a stable manner with no hydrophobic interaction, actually the toxicity, instead of being payload-like, now is a very particular toxicity of corneal epithelial cell toxicity. And you could think of those the corneal epithelial cells are the canary in the coal mine, at least for potent tubulin agents. Now then, there are also some pharmacokinetic considerations. Remember, some 99% of your ADC is going to be catabolized somewhere in the body, not much by the tumor. And this has consequences when one thinks of pharmacokinetic profiles. So just look at the top line here. This is the PK profile of exatecan. So in 2 days, the concentration in the blood plasma is 2.5 logs less than the starting dose. Now, same starting dose of exatecan, but now in conjugated form, it now has the PK profile of an antibody, except that it's slowly being removed and catabolized, which of course results in a continuous generation of deruxtecan in this case. Now, this is a log scale, so that's 100-fold lower concentration, but there's a constant concentration of exatecan for a long time, whereas this will, if you give the free drug, it becomes unmeasurable very quickly, because as fast as it's being removed, more is being formed. Similar story with dolastatin 10 and MMAE on an ADC, except that here the difference between the free drug and the payload is just a factor of 10, not a factor of 100, in part because of this generation of compound by the catabolism by somewhat hydrophobic species. Another emerging lesson from a lot of clinical data now is that while this was the initial idea behind ADCs, it turns out that the maximum tolerated dose of free drug and the maximum tolerated dose of conjugated drug in conjugated form is about the same. What the toxicities are may be very different, especially if this is a stable link, but you don't actually end up giving much more drug. However, it's on the efficacy side, essentially the minimum effective dose, that you get this great improvement of the ADC versus a free drug. Now, my final slide to introduce some later talks is there are multiple possible mechanisms for resistance to ADCs. There are antibody-resistant mechanisms. For example, loss of target. There was a poster this morning about reduction of HER2 in HER2-positive breast cancer, or alteration in intracellular trafficking, so less goes to the lysosome. And then, of course, you're releasing a free drug. So current ADCs are essential single-agent chemotherapy. And so the most likely mechanism of resistance in general is that payload resistance mechanisms common to classic chemotherapy. Why would you expect any difference? Now, one small difference would be ADCs with a non-cleavable link may be the exception to the above because of the absolute requirement for lysosomal processing, as shown here with TDM1. So if this was HER2, TDM1 binds, ends up in the endosome. A lot of that recycles, especially on HER2 low patients, so actually the trafficking to lysosomes is very poor. But on HER2 overexpressed, you get trafficking to lysosomes, complete degradation of the antibody to release DM1, still with the linker, and a lysine amino acid, the residue to which the whole thing was attached of the antibody. Now, this is charged, and so it doesn't get out of cells. But it does mean that resistance mechanisms, anything that interferes with this trafficking to lysosome, could affect TDM1 sensitivity. Whereas with cleavable links and diffusible payloads, the other thing to bear in mind, especially with the classic val-cit PAB, and it's probably true of gly-gly, phi-gly, a peptide link can be cleaved by cathepsin B, but cancer cells have cathepsin B in the endosome and secrete cathepsin B. So actually you can get release of payload in endosomes and also in the tumor microenvironment. So the resistance is more likely to be classic payload resistance mechanisms with a cleavable link and diffusible payload. So now there are plenty of tools in the toolbox to make ADCs from new linkers, payloads, improvements in conjugation chemistry. I think the targeting vehicles is going to be a next range of innovation. Most ADCs have been just IgG1s to address all of the different parameters to improve ADC. So with continuing innovation of the ADC design toolkit to improve tox profiles, the therapeutic index, and to address resistance mechanisms. So with that, there's far too many people in my 43-year career to thank individually. It takes teamwork to develop ADCs, so thank you very much.
Thank you so much, Dr. Lambert, for this fascinating and comprehensive review of where ADCs come from and how they work. And I think it's clear that they work in a complex manner, but when we are in the clinic, we need to translate that complexity into actual treatment decisions for our patients. And I cannot think of anybody better than Professor Curigliano to guide us through how we use ADCs in the clinic to treat breast cancer. He's head of drug development at the European Institute of Oncology in Milan, future ESMO president, great mentor for myself, and especially great expert in the field of ADCs. So please, Giuseppe.
So thank you very much for your really very kind introduction. So I will provide an overview of what we are doing in clinical practice, and then I will try to focus on some challenges for the future. So in 2013, there was only one ADC approved. It was TDM1. In the last 2 years, we have all patients within metastatic breast cancer or early breast cancer that can receive at least one ADC, triple negative, HR positive, HER2 positive. What is going on finally? In the HER2 positive disease, we located trastuzumab deruxtecan in the second line. We are going to move in the first line if the Destiny Breast 11 will be positive. And if you remember in the past, TDM1 was based on a clinical study in which a comparison with an antibody plus chemotherapy demonstrated an improvement in median progression-free survival and overall survival, and then moved to the adjuvant setting with the Catherine trial, in which we have both an invasive disease-free survival and an overall survival benefit. When the new generation ADC arrived, there was an optimization on the impact of these drugs in clinical practice. Just to remind to you that DESTINY-3, the median progression-free survival was 28 months. Just think that in the Cleopatra is 19 months. So what we are going to expect from the DESTINY-9 first-line trastuzumab deruxtecan, would it be 3 years of median PFS? So this is really the impact that these ADC have in our clinical practice. What's about activity in brain mets? You know perfectly, we discussed for many years, are ADC going to work in brain mets? Well, if you check the DESTINY-12, it is a real-world study, of course, with patients in a real-world setting, but you will see here that patients with measurable CNS disease have a response rate of 82%. That, in my opinion, it's quite impressive. You can delay radiation therapy if you have small brain mets asymptomatic. This is an important clinical message that is very clear for our disease where you have an oncogene addiction, but was not so clear for brain mets in metastatic breast cancer. And at 12 months, 61% of the patients are without brain progression, and also the data of overall survival, in my opinion, are impressive. This is a spatial population. What is next scenario? DESTINY-9, first-line trastuzumab deruxtecan versus the standard of care. This will open some questions. Should we give for 2 or 3 years trastuzumab deruxtecan to patients with HER2-positive disease? We know actually we stop chemotherapy. We give a maintenance with a dual blockade. That's why we have some maintenance trial. One is the Demeter, in which we stop TDX and we give a dual blockade, maybe with the opportunity also to rechallenge. Other trials are exploring the maintenance, but not in the post-TDX setting. One is the Patina, the second one is HER2CLIMB-05 with tucatinib, of course, plus pertuzumab and trastuzumab. What's about HR-positive? Actually, in HR-positive disease, trastuzumab deruxtecan is approved after at least 1 line of chemotherapy, and we have sacituzumab govitecan in the later setting. But we know perfectly that in the context of HER2-low disease, we also generated data on the HER2 ultra-low population. So with the lower expression of HER2, you can achieve still a clinical benefit. Don't forget that HER2 zero, according to ASCO-CAP, is not really zero. It's just a normal expression of HER2. So it's quite rare to have really HER2 zero tumors. Of course, in the DESTINY-6, the central assessment of HER2 zero samples discovered that finally 40% were HER2 ultra-low and 24% were HER2 low. So it's really essential to better assess the level of expression of the target, because if you miss the target, you will miss the opportunity to offer a drug that can impact on outcome. The first data have been presented in DESTINY-4, dramatic improvement in median progression-free survival and overall survival in a cohort of patients who received at least 2 lines or 3 lines of chemotherapy. If you remember inclusion criteria, here was for patients immunohistochemical 1+ and 2+ with FISH non-amplified. And then the data of DESTINY-6. The question behind DESTINY-6 is, can we anticipate the use of trastuzumab deruxtecan in patients who never received chemotherapy? And can we use it in their HER2 ultra-low population? I mean patients with a tumor with an expression of HER2 more than 0% and less than 10%. Dramatic improvement in median progression-free survival from 5 months to 13 months. Trend for overall survival, you will see this data next year. but it was surprising for us to discover that also in HER2 ultra-low there is an activity. So, of course, you can claim if this is really an effect, so-called bystander effect, or it is really an activity of trastuzumab deruxtecan with the minimal expression of the target antigen. Just yesterday, Aditya Bardia presented this very interesting data, in my opinion. In the DESTINY-6, there was a cohort of patients who progressed within 6 months after starting CDK4/6 inhibitors in the metastatic setting. The median progression-free survival, you can see here, is 14 months. So this is really a treatment option that should be considered also in patients with rapidly progressive disease, if HR positive, HER2 negative. And this may open also opportunity to consider first-line studies in those patients who will relapse during adjuvant CDK4/6 inhibitors. The second ADC was sacituzumab govitecan. These are the data of course, the TROPiCS-02 study, in which sacituzumab anti-TROP2 ADC has been tested in later lines of treatment, so after 2 or 3 lines of chemotherapy. Improvement of median progression-free survival and overall survival, and you saw also the data of datopotamab deruxtecan is a little bit different. The drug-antibody ratio is 1:4. There is an improvement in median PFS, but no improvement on overall survival. So not all the ADC targeting the same target antigen are the same. This is an example, of course. And what's about the data of patritumab deruxtecan? It's not approved, but I really believe the Icarus Breast 01 was really a seminal trial, because if you select patients and you treat with the clear translational study design, overall response rate is 53% and median PFS was 7 months with the lower incidence of ILD. This opened the opportunity, of course, to test the activity of patritumab deruxtecan also in similar setting of the other ADC. What's next? Sacituzumab timurotecan, and anticipating sacituzumab govitecan in the ASCENT-7 in early line of treatment. And finally, we have triple negative. Actually, we have 2 ADC approved. One is sacituzumab govitecan. The second one is trastuzumab deruxtecan in the setting of HER2-low. But we have under development datopotamab deruxtecan and sacituzumab timurotecan. In the ASCENT trial, sacituzumab govitecan was tested in later lines with an improvement of overall survival. And we had the data of sacituzumab timurotecan in the same patient population with a dramatic improvement in median progression-free survival and overall survival. Don't forget, these 2 ADC have a similar target, a similar payload, and we will check, of course, in prospective randomized trial, which will be the final activity. Another point is the study ongoing with datopotamab deruxtecan. We have data from the Tropion pan-tumor 01 with really interesting data of activity. And don't forget also the use of trastuzumab deruxtecan. Specifically in Europe, you know, there is a label for patients rapidly progressing after neoadjuvant adjuvant treatment, triple negative disease or HER2-low. So next will be moving anti-TROP2 ADC in the frontline setting, both as monotherapy in PD-L1 negative and in combination with immunocheckpoint inhibitors in patients PD-L1 positive. So which are the challenges finally? First, I believe ADC sequence. This is a major challenge. If you just check the data from prospective randomized trial, if you sequence in the DESTINY-2 after T-DM1 trastuzumab deruxtecan, there is a dramatic improvement in median PFS. So we have 2 ADC targeting the same target antigen, but with a different payload. And here you can see the activity. But in all the real-world studies, the second ADC has a shorter median progression-free survival respect to the first ADC. In this figure is very clear. This is the first ADC, this is the second ADC, and the median PFS is completely different. Is this related to mechanism of resistance? This is a beautiful study, I believe, in which patients receiving sacituzumab govitecan have been biopsied on the sites of progressive disease, and investigators discovered mutation in topo1, so in the enzyme metabolizing the payload, and also mutations in the TROP2, in the binding ligand. So it seems that if you have a concurrent mutation on TOP1 and TROP2, the binding antigen and the enzyme metabolizing, of course, the payload, you can develop, of course, a mechanism of resistance. So it means mechanism of resistance can be related to target antigen, can be related to the payload. And you know perfectly that the question on sequencing are related to same antibody target or same mechanism of action of the payload. I would never sequence similar payload. The use in sequence of two ADC sharing the same target but different payload may overcome this mechanism of resistance. Less data exists on the use of a sequence of two ADC with different targets, but with similar mechanism of action. We don't have this data, and we need to develop. And confirmation of significant cross-resistance between these ADCs would reinforce the need for payload differentiation. Up to now, the majority of ADC are conjugated with topo1 inhibitors. Second challenge are toxicities. This is a challenge, of course. We have several ADC with different safety profile. Within the same class, we have anti-TROP2 that will give diarrhea, and we have anti-TROP2 with mucositis. How can we explain this? We really don't know absolutely nothing on this. But we know perfectly that when you use an ADC, you have an on-target toxicity and you have an off-tumor toxicity. Specifically, if you use enfortumab vedotin, of course, you will have skin toxicity because Nectin-4 is overexpressed in the skin. For tissue factor ADC, you will have ocular toxicity. And for HER2, you know perfectly that some macrophages can express HER2 within the lung, and this can be a potential explanation. Another mechanism of toxicity is related to the payload. One thing is if you use a microtubule inhibitor. Another thing is if you use a topo1 inhibitor, and you know perfectly that toxicity can be related in the first scenario, neurotoxicity. In the second one, GI symptoms, of course, fatigue and neutropenia. Don't forget that only 1% of the payload ADC will reach the tumor bed. This is really impressive. I believe that maybe in the future, with development of theranostic, I am quite sure we will be able to better understand how much drug can reach the target. Because if you perform at the baseline and molecular imaging, and then you perform after treatment, if you discover that you kill at the positive cells, this will better give us an opportunity to understand the mechanism of action. And finally, pharmacogenomics. This is a beautiful analysis on the ASCENT trial, a specific polymorphism that is UGT1A1 2828. This is completely absent in the Asian population, but in Caucasian population is very frequent, and this is associated with more toxicity, more neutropenia, less time of onset for febrile neutropenia. So how to predict toxicity of ADC? It's a multidimensional evaluation, including patient characteristics, body mass index, third space fluids, age, sex, ethnicity, polymorphism, concomitant medication, then prior therapies, if you have any residual toxicity, toxicity timelines, tumor location, in my opinion, and then, of course, we need dose adjustment and optimization, and we need to better understand which are the delayed effects of chemotherapy. The final challenge are biomarkers. So antibodies are targeting a biomarker. So we have some antibodies approved based on a biomarker expression and some others without a biomarker expression. Just if we check the DAISY trial, it's a very simple trial evaluating the activity of trastuzumab deruxtecan in 3+, 1 or 2+, and 0. Here there is no definition of ultra-low. And you can see here overall response rate, ranges from 30% in HER2-0, 37% in HER2-low, and 70% in HER2-3+. Just think about the hazard ratio. Destiny-3, the hazard ratio is 0.3. In the Destiny-4, is close to 0.5. In the Destiny-6, it's 0.6 if you check in the population with their HER2-low disease, but it's 0.7 in their HER2-ultra-low population. So it means that increasing expression of the target may predict the response rate and benefit in terms of median PFS. And this is exactly the same also for sacituzumab govitecan, of course, is a subgroup analysis. We don't need to assess TROP2 for triple negative, but there is a clear difference if you have a high TROP2 and low TROP2. What's about mRNA? This is a very nice study on mRNA expression. Seems to be no association between RNA-seq and eventually level of RNA and the predicting activity of sacituzumab govitecan. So future opportunities, these are the last slides. I believe personally that in any new development of an ADC, mainly in the phase 1, you need to do more biomarker research. So you need to understand if any biomarker can predict the activity of the ADC. This is a very interesting analysis with a quantitative continuous score, a next generation target to understand heterogeneity in the context of a tumor, that of course can better select beyond the human capability the number of cells and the spatial distribution of cells that can really predict the activity of trastuzumab deruxtecan. So this quickly identify with the proteomic analysis and the visual analysis imaging, multiplexed proteins for single sample quantification that directly are comparable by absolute quantification, much more than the human eye. And if you check the data, this quantitative continuous score can really better predict the patients who derive more benefit from trastuzumab deruxtecan. I don't know if this type of technology can be applied for any other ADC, but if you just remember, for enfortumab vedotin, if you have an amplification of Nectin-4, you may have much more benefit from the ADC. And similar for sacituzumab govitecan in the context of the ASCENT trial. So in conclusion, I really believe that ADCs have reshaped the treatment approach for patients with metastatic breast cancer in HR-positive, triple-negative, and HER2-positive disease. Given the concurrent development of multiple agents, the best ADC sequencing, in my opinion, remains unclear, and we need to design new clinical trials of sequencing. Sequencing doesn't mean you give a first ADC, and once you progress, you give the second ADC. You can do a sequence like we did sequencing anthracycline and taxanes in the 80s. This is the ideal study design to better understand if median PFS2 is better than median PFS with a single ADC. Biomarker of resistance and activity are needed to personalize treatment, and in my opinion, effective toxicity prevention, monitoring, and management are essential to maximize the risk-benefit ratio. Thank you very much to all the people that helped me in this presentation.
Thank you, Giuseppe. This was an outstanding review of how to use ADCs in clinical practice. And I think it's clear that they really have reshaped outcomes for metastatic breast cancer, and they're continuing to do so. We're learning every day how to use them better. I will remind everyone that you can submit questions online, and also at the end, we will have a question and answer, and we have microphones in the room. But I think another thing we learned in the past decades is really that it takes a village to understand how to use ADCs and to develop ADCs. A village both that includes academia and industry. And I think it's important to understand from somebody that has been in both how this works. How do you actually develop an ADC, take it to the clinic, and choose which ADC to use in which indication. And so I would like to invite Ingrid Mayer, Global Head of Strategy for AstraZeneca, to guide us through her path and also what she learned in academia and industry for development of ADCs. Please, Ingrid.
Thank you. So, hello everyone, welcome to San Antonio. So it's let me start by asking, like, why the hell am I here? So it's a fun fact where Paolo invited me, and I wasn't sure, what the remit of my talk is, and clearly, I still don't know what the remit of my talk clearly is, and I'll try to do like a snippet of what that is, because there's clearly going to be a lot of overlap between all of our talks, as expected. But nevertheless, let me start by acknowledging Pooja Sapra, who works with me. She actually is a wizard in developing ADCs, and she helped me put this talk together. And hopefully I'll bring some of the color of what I have learned in the industry since transitioning from academia. And with that, let me start by telling you a little bit about my journey. So I was happy as a clam, minding my own business at Vanderbilt for almost 20 years, between 2003 and 2021. Towards the end of my career there, I was the head of the breast cancer program and obviously very involved with clinical trials for all sorts of indications in breast cancer, all sorts of drugs and whatnot, and ADCs really have taken a lot of flight at that point. So an opportunity knocked on my door, kind of like in the middle of COVID. I had a midlife crisis and decided to transition to industry to potentially have a much bigger impact in what we can do for breast cancer patients, much more so than I felt I could do in academia. So there I was, I transitioned at the end of 2021. Literally, I moved to industry 1 week before San Antonio in 2021. So you can't imagine what that San Antonio will look like for me. And for a good 6 months, I think I was like deer in the headlights, like not understanding what the heck did I do? Because it was a brand new world and very, very different than how we do things in academia for a variety of different reasons, specifically for clinical trial design. So while drinking for a fire hose for a whole 6 months, after that went through, I realized that instead of transitioning to the dark side, as we usually say in academia, I think I've transitioned to the bright side because it has been an incredibly fulfilling experience, incredibly intellectually challenging, and really very fulfilling in terms of the magnitude of benefit that we can promote by bringing new drugs to patients in a way that affects patients not only regionally, which what we typically do in academic centers with phase 1 and phase 2 trials, but globally, where it's beyond just bringing drugs, it's also breaking barriers to clinical trial participation and breaking barriers to access to drugs. So with that, let me focus on the premise of ADCs. You've heard this before. I'm not going to spend a long time on this slide other than to say that we really do acknowledge that ADCs bring really a novelty compared to what chemotherapy can bring, but the stars have to align correctly for ADCs to work. You need to have a great target, you need to have a great linker, you need to have internalization. Hopefully, if you're lucky, you get bystander effect. Hopefully you can develop some immunogenicity in the tumor microenvironment, which is very conducive for combinations with immunotherapy. So there's a lot of things that need to happen in a correct way to hopefully get us to that perfect ADC that would work well and really elevate outcomes on top of chemotherapy with a very favorable therapeutic index. So how does things different in terms of how we develop drugs in general, but specifically ADCs in academia? So from my experience, what we usually chose as control arm when we were designing a trial had a very regional flavor. Obviously, I worked in the US, so I usually would flock naturally to what control arms would be in the US. But that really didn't give me a sense that, you know what, maybe in Europe people do things differently. Maybe in Asia they do it differently. There are parts of the world that they don't even have access to those drugs. So sometimes when we design something in academia, we are not looking at things at a global level. The other thing that is quite striking is when you're designing phase 2s, randomized phase 2s, or even phase 3s in a cooperative group, you actually have a little bit more flexibility in your trial design where you tend to ask multiple questions within a trial, which is something that we really shouldn't be doing, but nevertheless is tempting. And we have a lot more flexibility with the statistical design where we're more permissive, whatnot, because there's no pressure that we need to take that trial for a registrational approval. And therefore, again, we tend to be more permissive, more flexible, and so forth. The trials tend to be smaller because we usually depend on budget that would either come from grants or pharma support and whatnot, and that money really is finite. And therefore, we are really not able to do trials that are great, and we end up sacrificing some statistical integrity in order to achieve that. And lastly, as in academia, obviously you do something that is very disease-specific, and you're not really thinking about the development of that drug in what other indications can that ADC or literally any other drug reach. So when you move to industry, there's a whole new world of things that now you have to consider when you're developing ADCs. You have to consider who your patient population of interest is. What is the control arm, the trial design, the contribution of components. Like that term was not even in my radar when I was in academia. Contribution of components, what the hell is that? But that's actually critical because you really need to prove on different drugs, especially when you're doing combination, which drug is actually providing what benefit, and is the added toxicity justifiable? Contribution of phases, dose optimization, all the statistical considerations, all of that needs to be globally friendly to multiple regulatory agencies, not just the FDA or EMA, but literally everywhere. We need to consider different biomarker cutoffs. I mean, you heard how important the target is and the expression of the target. So when you're developing an ADC and you want to maximize that efficacy, how do you choose what the optimal cutoff of that ADC is? Because from a corporate point of view, we want this to be as broad as possible. But is that actually the right thing to do? Like how low can you go or how high should you go? We need to consider the development of companion diagnostic markers, which again is not something that ever crossed my mind in academia, that you actually need to develop a whole test that goes along with it and needs to be approved as well by regulatory agencies. You need to consider, again, if you're going to bring this to other indications, what treatment settings are you going to bring this in? Because we know that on a patient journey, things behave differently in the early setting than in metastatic setting. We need to consider the merits of potentially having different formulations, like sub-Q formulations or IV or anything else. And this one blew my mind. Like, what the hell is pediatric development? Like, why are we bringing certain drugs in pediatric patients? But actually, that is a thing, because there are pediatric diseases, pediatric cancers, that may benefit from some of the drugs that actually are more relevant for adult solid tumors. But nevertheless, you do need to have a whole pediatric plan scheduled out for literally every drug that you develop. Then you have the whole commercial issue of how do you distribute these drugs, how do you manufacture these drugs, how long is it going to take to get that done? You need to consider what label do you want, how do you commercialize this, what is the reimbursement going to be on a global level, and finally, all our competitors in the field, like how much of a market share are we going to get, like what are the competitors doing, how fast do we need to go, what corners do we need to cut, and whatnot. So all of these are things that we really, really need to pay attention. And I swear that I probably didn't list a whole bunch of other things that we need to take into account when we think about clinical trials for ADC developed in academia, in industry. But the one thing that is true for both worlds, and I'm going to stress that out, is that always, always, always follow the science. Always follow the biology of the disease. I cannot emphasize how important that is, because if you don't, you're essentially going to bring the wrong drug to the wrong patient at the wrong time, and you're going to have a failed trial. So you really need to understand the disease biology of where you're going after, and you need to understand the performance of your drug and how far can you go in pushing that drug. And ultimately, from a patient perspective, it's really ever, always about benefit-risk ratio. Because as I said before, sometimes when you're doing combination trials or even when you're bringing a new drug, you need to have that healthy balance between potential added toxicity or not, and how much efficacy needs to happen to justify that added toxicity or not. So that's, I think, the big take-home of my talk, is where we meet is really doing the right thing for patients and following the science at all times. And that is really the big intersection between academia and industry. So the rest of my talk is going to have a substantial overlap with the first two speakers and probably with the third speaker, so I apologize for that. But you know what my parents always say, repetition is the mother of learning. So there you go. You're going to hear things said in a different way, but hopefully you'll take it home, some of the message that all of us try to impart today. So from an ADC development, obviously there are things we need to consider. The targets are something important we need to consider. We need to consider the conjugation of all the different elements that make up that ADC and the payload. So let's go over some of the key things that are important to consider for each one of these individual elements. So from a target point of view, how do we choose the best target? So Professor Lambert said that, Professor Curigliano said that, I'm going to say it again. look at the biology. I cannot emphasize that enough. Understand the disease you're taking that ADC to. So additional activity at ADCs are important there. You need to understand the surface protein density of the target you're trying to hit. You need to make sure that the ADC is going to internalize effectively. You need to make sure that your target is not or is heterogeneity expressed, and how does your bystander effect circumvent that. And most importantly, you need to understand what the normal tissue expression of that target is, because that, I can guarantee you, is going to bring a lot of off-target toxicity. So from a target point of view, I think we have moved away from looking at the targets either by RNA or protein measurements, and now have moved to an era where we can take advantage of proteomics to actually detect what is the best target that we can go after. And that gives you a much more accurate depiction of what targets are more prevalent and better suited for an ADC. The other cool thing we can do with ADCs is take advantage of what we call co-expression of targets. So if you imagine EGFR as a target, and to bring this to the breast cancer world, as you know, triple-negative breast cancer has an incredible expression of EGFR. But EGFR is also expressed pretty much everywhere else in your body. And if you were to deploy a very effective ADC against EGFR, your off-target toxicity outside the tumor would be enormous. But when you co-localize that expression with a c-Met expression, which in your skin, you don't get EGFR and c-Met. You get c-Met in some places, EGFR in other places, but not necessarily both. But in the tumor, you do. And therefore, by co-localizing this target, you actually can get your ADC to attack the tumor, but not necessarily some of the tissues that express one or the other target. You need to express both for the ADC to work. So that is the premise of potentially new development of ADCs that co-localize different targets, taking advantage of that and minimizing the off-target toxicity. Again, the site-specific conjugation, that has to do with the drug-antibody ratio that you put in there. Depending on how you put that in your molecule, and please do not ask me how they do that. Dr. Lambert would probably be able to explain that better than I can. But nevertheless, that does make a difference. And depending on how you do, you can actually, again, minimize the toxicity you provide by being able to be more efficient in your DAR expression and more stability in your payload release. In terms of payloads, I think, again, Dr. Lambert and Dr. Curigliano did a phenomenal job already guiding us through the different ones that went through there. But let's focus on the last box in there, not to repeat what they already said, where I think we now are in a good position to explore things beyond microtubular inhibitors and beyond topo-1 inhibitors, because topo-1 inhibitors, they are super active, but now it's gotten old, and we can't just recycle payload after payload using the same mechanism of action. And microtubule inhibitors, unfortunately, suffer from neuropathy as of those limiting side effects. Therefore, things like innate immune agonists, PROTACs, DDR inhibitors, epigenetic modifiers, or cell-death inhibitors could actually be new payloads that could allow us to sequence ADCs in a very efficient way, where we still have superior efficacy over chemo, but we're not recycling the same payload. So with HER2 and trastuzumab deruxtecan, I think this was a very, I wouldn't call it a happy coincidence. I'm sure there's a lot of work that went behind that. But HER2 and trastuzumab deruxtecan are about as perfect as an ADC gets today. And hopefully we can build on that and learn from what went right in the development of that. So two things that I think I call out as being quite the perfect storm in there in reverse, right? The first one is the fact that the target is a very happy choice, because the target here is an oncogenic driver. So it's not just about the conduit to get something to link in there, internalize, and exert the cytotoxic effect, like TROP2 is, or maybe Nectin-4 or whatnot. This actually has biological relevance. You actually get some effect just by binding onto HER2 and suppressing that oncogenic driver. So I think that was a happy, not coincidence, but a happy thought on how trastuzumab deruxtecan was developed. Topoisomerase 1 payload obviously are very active in a variety of breast cancers, and therefore both HER2, HER2 positive, and triple negative, HER2 positive, ER positive, and triple negative disease all benefit from topo-1 isomerase payloads, because that's not typically an MOA that we normally use. Like, it's not that dissimilar than what anthracyclines, platinum agents, whatnot work against the tumor. It's all about DNA damage, but nevertheless, it's still a little bit different than these other MOAs, and this was kind of like new and refreshing from a breast cancer point of view. Trastuzumab deruxtecan has a high drug-antibody ratio, it has a very stable linker, and it has bystander effect. So I think I would argue that trastuzumab deruxtecan is, I wouldn't say an outlier, but maybe it is a little bit of an outlier. It is a very, very high bar to beat, but that really just needs to be our North Star of how are we going to develop the next wave of ADC or ADC-like compounds. So from a company perspective, what are the areas that we need to invest? And again, you've heard this from speakers. Biomarkers is an important one, and how do we better detect these biomarkers? Leveraging artificial intelligence to do so. Really banking on mechanistic combinations, especially with IO or other DNA damaging agents, whatnot, because at the end of the day, from a cancer point of view, it's all about attacking the cancer through multiple angles. We need to outsmart the cancer and taking advantage of the potential synergy that we can see from ADCs and immunotherapy or other agents. It's something that we need to consider as we are developing how do we want to bring these ADCs into the clinic. And lastly, as pointed out by Dr. Curigliano and Dr. Lambert, understanding resistance is obviously key. So digital pathology, again, I'm not going to spend a long time on this. Dr. Curigliano already mentioned that leveraging this quantitative computational score, it's something that we are hell-bent to do at AstraZeneca, where we take us from just using IHC, which is imperfect and relies on someone's eyes, to something that is a little bit more reliable and hopefully reproducible that could actually take the understanding of which patient population an ADC goes, and again, that concept of how low can you go on the cutoff of that ADC. QCS is really probably something that is going to get us there. In terms of understanding the target expression, I go back to the biology. It's not just about the prevalence of the target, but it's also, does that prevalence of the target match a disease that is sensitive to the payload of that ADC? Because, as mentioned before, if you have an MTI ADC that has, just to use the EGFR example before, EGFR expression, and you want to put it against colon cancer, would that work? The binding would work, but does MTI really work in colon cancer? Not that much. I mean, it's not 5FU, right? So I think you just need to kind of like connect the dots and make sure that your payload matches the biology, and that particular disease has the expression of the target you need to have. And understanding the disease biology also allows you to think about creative combinations that could enhance what that ADC is doing. So using, for instance, diseases that have dependency on DNA damage repair, either BRCA mutated cancers or PALB2 mutated cancers, or even cancers with homologous recombination deficiency, perhaps combining an ADC with a PARP inhibitor, even though the mechanisms of actions are similar in terms of DNA damage to some extent, one could leverage a small dose of PARP inhibition to promote an increased replication stress, and in doing so, perhaps you could prolong how long an ADC would work. And then, again, this mechanistic combination of IO and ADCs is also a happy one, because ADCs have the potential to prime the tumor, to prime the microenvironment of the tumor to be more immunogenic. And also, the T-cell depletion with ADCs is potentially not as strong as we see with chemo, because some of them don't have as much overall myelosuppression as chemotherapy agents do. So with that, we already see live proof of that, of these combinations, not just in breast cancer. We clearly see synergy between trastuzumab deruxtecan and durvalumab, with datopotamab deruxtecan and durvalumab, Nectin-4 ADCs with Pembro. So all that is already a reality in the clinic, and I think we're going to see more and more and more of these combinations of IO and ADCs coming to fruition. Resistance is something that also has been mentioned before. I'm not going to again spend too much time on this slide because all the other speakers already went through that, but I'm going to have you focus on the payload as a mechanism of resistance, because while all of these mechanisms listed there, the target, the antibody, the linker, and the payload, everything can go wrong in there, but if I were to bet money on what is the most likely reason for why a tumor becomes resistant to an ADC, I would bet my money on the payload. And therefore, doing exactly what Dr. Curigliano says, sequencing ADCs with different payloads, it's something that is a very obvious thing to do, and we know it works. I have 2 examples on the slides on that. TDXD after TDM1 is one, FR-alpha MTI ADC followed by an FR-alpha topo-1 ADC is another one. And really understanding that, I think, would help us craft how should a patient journey leverage different ADCs that can be sequenced or even combined in a smart way. So going back to the clinical development considerations, again, I think the overarching goal for us from an industry perspective is to bring novel drugs to the global market ASAP. And while that actually sounds like a very corporate goal, believe me, it's actually not. Because while we need to ensure there's global registration success and reimbursement, whatnot, the ultimate so what, and so what is a phrase we use a lot, the ultimate so what is because bringing these drugs to market sooner actually bring transformational outcomes for patients sooner. So it's really not about the money, it's really about making an impact and bringing drugs that work to patients in a much more efficient way. And while we have to juggle a lot of balls in the air, like the ones that I have depicted in there, of all these things that we have to take into account, again, I cannot stress enough of the importance of having us follow the science and the biology and acknowledging the benefit and risk. So where can the future take us? Well, I think, again, from a payload and linker point of view and novel target discovery, we can improve on what we've done today. From a biomarker point of view, better detection, leveraging QCS technology can get us there. And novel ADC combination and sequencing also can get us there. In terms of expanding beyond cytotoxics, I think a new wave of ADCs that may include radioconjugates, it's something really cool to look forward to. Immune-stimulating antibody conjugates, antibodies that use PROTACs in the payload, and a whole variety of additional modalities as well. Many questions still remain on how can we improve that. So as I said, I think enabling ADCs beyond classic cytotoxics, how do we do that? How do we overcome off-target tox issues? How do we enable multiplex diagnostics? Can we use blood-based diagnostics, like including ctDNA, to better select prediction of benefit? Can we design mathematical model to capture the complexity of ADCs? And what are the mechanisms of clinical resistance? And can we actually use ADCs beyond oncology? But I think for now, I think ADCs are definitely here to stay. I'm so happy to see them in the clinic, really making a significant mark on patient survival. And again, all these innovations, I think, are going to continue, and this is going to be a very, very exciting decade coming forward. But last phrase, and I promise it's the last time I say this, but again, deep understanding of disease biology is key for the clinical development of an ADC, and matching the right patient to the right drug at the right time really is where academia and industry intersect in our interests in drug development. So with that, thank you. And
thank you, Dr. Mayer, for that outstanding presentation, which I think sets perfectly the stage to talk about the future. Because in recent years, we've seen very rapidly things changing in the ADC field, but I believe it's just starting. And I have some proof for that belief. So I'm going to talk about how to aim for the target in the future through novel molecular constructs, novel biomarkers, and in general, novel ideas. These are my disclosures. And so the proof is basically that, as we've seen, as Dr. Lambert has presented, it took 20 years from the early 80s to have the first approval of an ADC in 2000. And then it took them more than 10 years to have the second ADC approved, TDM1. But then, by contrast, in the last 5 years, 8 novel ADCs were approved. And where will the next 5 years take us? I think it will take us to have many more ADCs, but also many more challenges, as we were seeing presented by Professor Curigliano. So let's look at it through the future cons, basically a concept that was developed when the ADCs were starting to be developed in the 80s by the futurists. And the way to look at them is that if the broader the cone, the less likely, the more broad is the possibilities. The narrower the cone, instead, it means that it's most likely to happen. And so one of the futures that was theorized by the futurists is the probable future, the business as usual future. And so in the ADC field, we have right now 3 ADCs that we can use: TDM1, TDXT, sacituzumab govitecan. 2 more are coming: darapotamab deruxtecan. There's a BLA submitted for HR-positive disease. Sacituzumab tirumotecan already approved in China, and we may see several phase 3 trials reading out in the future. And so when you look at the trials, phase 3 trials that are ongoing in the ADC field, these only in Europe and in the US, you have several trials in HER2-positive disease, multiple trials in the triple-negative disease in green, and also in HR-positive disease. All of these are in the neoadjuvant, adjuvant, first-line cytotoxic setting, so earlier than we are using them right now. And one thing that all of these trials have in common is that all of the trials involve topo-1 ADCs, come to test in payloads. And another thing they have in common, as I mentioned, is that they are in early line. So I think the business as usual future for ADCs is that we're gonna have more topo-1 ADCs, and they're gonna come in earlier indications in the first line, and hopefully they're gonna be able also to cure patients in the neoadjuvant and adjuvant setting. But this, as we've seen, comes with challenges. We have seen that one key challenge is sequencing. I got the chance to present this poster today, which basically found what many real-world analyses are finding, that if after a topo-1 ADC, you use another topo-1 ADC, you tend to have less than 3 months of PFS, but if you switch around the mechanism of action with something like capecitabine or eribulin, actually you can get more than double than that. And so probably we have to start thinking about, of course, differentiating payload mechanisms of action, as also Dr. Mayer has mentioned. But at the same time, this is all retrospective evidence. We need prospective evidence, and I'm glad for all the investigators and companies that are working at this. At Dana-Farber, we are working with 2 key trials, with the SATIN phase 2 trial. There is the first study that tests sacituzumab govitecan in HER2-positive metastatic breast cancer combined with trastuzumab, of course with external collaborators, and TRADE-DXT. That is the first randomized ADC sequencing trial testing TDXT after Dato-DXT or vice versa. And hopefully these trials will provide some more data in the field about sequencing. And I wanted to acknowledge one major personality that developed deruxtecan and unfortunately passed 2 days ago, Toshinori Agatsuma from Daiichi, that led the first studies and really had a major impact in the field. And it was a big loss for the field of ADCs, I feel. But let's move to another type of future, the plausible future. So we said there's many phase 3 trials ongoing, but there's also many phase 1 and 2 trials ongoing for ADCs. And one key thing that is important, I think, about this class of agents is that they're modular. They're like Lego. You can basically switch around one of the 3 key components, the antibody, the linker, or the payload, and you can get a completely different clinical activity. We've seen it with TDM1 and TDXT. Same identical antibody, but by switching linker and payload, you have a 4-fold improvement in PFS. And all of this is still ongoing. We are learning how to optimize and how to innovate the antibody, for instance, through bispecific antibodies. What they potentially can do is increase the internalization rate of the ADC. They can expand the patient population, so you can target a tumor that expresses antigen 1 or 2, or increase selectivity, so target a tumor that expresses both antigen 1 and 2. And there's several bispecific ADCs being developed. We've seen already some phase 1 trials reporting impressive response rate with bispecific ADCs, and all of this is moving very fast. There's many in the pipeline that are being tested in these days. But then we can also switch the antibody altogether and make it a small molecule, so to increase penetration in the tumor microenvironment. And bicycle toxin conjugates are attempting to do that with a much smaller molecule compared to an antibody, a bicycle that has a deeper tissue penetration. And we have already some data with an MMA bicycle toxin conjugate. And also we have some data with a peptide drug conjugate. Same idea. You have a much smaller molecule that increases penetration, also changes, of course, the PK, and so we're not sure if we're going to achieve the same activity or potentially more, but I do think that we need to be open-minded and test novel approaches like this. And interestingly, AVA 6000 releases doxorubicin, that we still don't have any ADC with doxorubicin in the clinic, and it can be specifically cleaved in the tumor microenvironment. So I think all of this may lead to innovation in the antibody. At the same time, we've seen how the stability of ADCs is key, and we can improve the stability of ADCs. In particular, if you think that all of the ADCs currently approved are unstable with different levels and types of instability, but most of the chemotherapy that is linked to the antibody gets released after a few days continuously after you dose each ADC. And so we can change that, we can modulate that by modulating the linking of the payload to the antibody. And all of the current ADC utilize what we call stochastic linking, meaning that the chemotherapy is basically linked quite randomly to either lysines or to other sites in the antibody, but in truth, we can improve the stability of these ADCs by making site-specific conjugation, introducing non-natural amino acids into the antibody, increasing let's say, the predictability of the PK of ADCs and potentially the stability, the delivery to the tumor of the chemotherapy. Finally, the payload. We've discussed about how important it is to differentiate the payload of ADCs and how the resistance of tumors to ADCs is largely dependent on the payload based on what we are learning. And still, of the more than 200 ADCs currently in the clinic, over 100, over half, have topo-1 inhibitors as payload. About 60 have microtubule inhibitors. A few have novel agents, or at least partly novel agents. For instance, we still don't have in the clinic an eribulin ADC, and we know that eribulin is an active agent. There is an ADC with an eribulin payload that showed 70% response rate for metastatic breast cancer, both HER2 positive and HER2 low. And right now, there is a phase 2 trial post-TDXT to see what is the response rate and activity of this agent post-TDXT. And I think it's fundamental to understand what is the activity of novel ADCs after agents like TDXT, sacituzumab govitecan, and currently approved ADCs. But in truth, I think we can innovate further. Why stick into just one chemotherapy when you can achieve a combined chemotherapy, a multi-chemotherapy within the same ADCs? And there's dual payload ADCs that are approaching the clinic and that can potentially achieve what we do, for instance, in the adjuvant setting, where we combine anthracyclines, taxane, platinum compounds, and potentially within an ADC, and it's coming. And then, of course, immune-stimulating ADCs. We know that even the current generation of ADCs is somewhat immune stimulating, but can we improve that by delivering a toll-like receptor agonist, for instance? And we already have some phase 1 data for this strategy. What about changing altogether and instead of looking at cytotoxicity, trying to degrade a specific protein in the tumor cell? Antibody-protein conjugates are also in the clinic right now being tested in phase 1, and finally radioimmunoconjugates. All of this just to say that really we can deliver potentially anything to the tumor, to the ADC technology, but it's important to always remember how ADCs work, because as we mentioned several times, only a small part of the ADC ends up in the tumor. So whatever you're going to link to the antibody may end up also in normal tissues, and this is important to be remembered with all of these novel ADCs. Then we have the might happen future. And I call it might happen not because I don't think it's going to happen, but just because it's still retrospective for the moment, most of the data that we have. But I do think that we need to study this prospectively. And I'm talking of novel biomarkers. Because in truth, we would expect, based on how ADCs work, that the more antigen is expressed in the tumor cell, the more activity you have with ADCs. And we've seen that this is true in certain instances, but not always. We had surprises. And so for several ADCs, the IHC expression of the target does not predict the activity of the ADCs. Some interesting examples were, for instance, DESTINY-Breast04. We were expecting in DB04 more activity with TDXT in HER2 2+, but in truth, it had the same activity in 1+ and 2+. Numerically, the PFS is slightly longer in 1+. And in DB06, there are some trends related to the hazard ratio, but in truth, TDXT worked amazingly also in HER2 0 breast cancer. What about TROPiCS-02? TROP2 highly expressing tumors and TROP2 lowly expressing tumors, they had the same overall survival benefit with sacituzumab govitecan over chemo, which is why we don't assess TROP2 in the clinic right now. So it's clear that IHC is not the ideal biomarker or assay to look for ADC's expression, which makes sense because we started looking, for instance, at HER2 IHC expression over 20 years ago, just to predict which patients benefit to trastuzumab, but not ADCs. And if you look at, for instance, the difference between IHC 0 and 1+, Well, it's not that easy to find the difference between these two slides. And you can expect some heterogeneity among pathologists reading these two slides into HER2 0, 1+, or the new category of HER2 ultra low. So one thing we're trying to do, for instance, at Dana-Farber to overcome this is to collect biopsies and plasma samples for patients that receive ADCs in the clinic before and after, and hopefully, and when possible, during treatment with ADCs, and utilizing a broad panel of novel assays to understand which may be the assay of the future or the assays of the future. And one thing we found is that a more sensitive detection of HER2 with quantitative immunofluorescence was granularly able to predict time to next treatment, which is a proxy for PFS with T-DXd in about 50 patients treated at Dana-Farber with T-DXd. And now we're expanding this cohort. And also we found that RPPA, another quantitative sensitive method to look at protein expression for HER2, nicely predicted the expression, the activity of trastuzumab deruxtecan in the clinic. And since RPPA can also assess quantitatively several other targets, we even looked at the payload target, topo1. And what we found interestingly is that tumors that express more topo1 seem to be more resistant to T-DXd. This was based on small numbers, and we're open to expand this, but it could potentially make sense that tumors that have more topo1 expression need additional doses of an ADC to be inhibited. And finally, we've seen some data in these days with HER2-DX, and it totally makes sense to think also at an mRNA assay that looks at HER2 expression or HER2 amplicon expression in terms of mRNA, and we did find in our cohort at Dana-Farber that this predicted outcomes with T-DXd. So we have many potential assays that may improve the way we tailor treatments in the future with ADCs, which will become more and more important when these agents move to the early setting, to the curative setting. Finally, there is another particular future that is called the preferable future, the should happen future. And I think in the field of ADCs, this relates to a toxicity profile of ADCs. We know that all ADCs have got side effects. They got side effects that relate to different parts of the ADC. And I also would like to highlight that there is an upcoming survey from patient advocates coordinated by Giulia Mayos that will come next year. And please participate if you're a patient advocate next year. But talking about why we have this toxicity, and we've heard some of this. It's heterogeneous, the reason. We have some toxicity because of unstable linkers that release payload in the circulation, because of target engagement, cardiotoxicity with trastuzumab deruxtecan, or skin toxicity with enfortumab vedotin. But in truth, usually the maximum tolerated dose of ADCs is related to the payload of ADCs, as we've seen. And so it's important to always consider the payload and to expect certain toxicities related to the payload. And ways we can limit these toxicities or optimize the use of ADCs is, for instance, dose capping. Some ADCs start becoming more toxic when you approach very high dose. And so in patients that have higher BMI, limit the dose to a certain cap. Capping of duration. We do this, for instance, with Taxol. You don't give it forever. You stop at a certain number of cycles. And with some ADCs, this is already done, like polatuzumab vedotin. It maybe need to be done more with some ADCs. Response-guided dosing. Now, right now, we use ADCs indefinitely, irrespective of if the patient is deriving a complete response, partial response, or stability. But should we start adapting, thinking about maybe reducing the dose of the ADC in patients in complete response, or starting a maintenance strategy? And all of this is already happening, and I think needs to happen even more. And fractionated dosing. We've seen gemtuzumab osagamicin, the first ADC to be ever approved. It was withdrawn by Pfizer, and then reintroduced after a decade with a fractionated dosing. And so thinking about the dosing and ways to make ADCs more tolerable, I think is critical. And then it's important to innovate the structure, the molecular structure of ADCs, and ways where they are being attempted to do so is by masking the antibody. And so you add the peptide mask with what we call pro-body drug conjugate that doesn't allow the ADC to bind to healthy tissues, but once the tumor microenvironment, the mask gets released by local enzymes in the tumor microenvironment and is able to link to a tumor. We already have some phase 1 data with these agents that still seem to have payload-related toxicity, as you may expect, but there is a lot going on in this field in general. And then, since we said that the payload is so fundamental for toxicity of ADCs, why not trying to block the payload that gets spilled in the circulation? And so there is another, let's say, field of research that is looking at payload binding fragments. So there are fragments that bind the payload that gets released from the ADC in the circulation, make it easily metabolized by the kidney, but they do not instead do the same in the tumor microenvironment. They allow the payload to diffuse easily in the tumor microenvironment. And we saw in preclinical models that this works well. It retains the activity of the ADC, but it prolongs and it leads to less weight loss in mice models. And so all of these strategies may help to make ADCs more tolerable. But I think overall, a very interesting movement that ADCs are having is that they're moving beyond oncology. And we are all oncologists or contributing to the oncology field. And so of course, we want to cure cancer, but there's many diseases in the world. And if we can leverage what we learned in oncology to also cure neurological diseases, rare diseases, immunological diseases, infectious diseases, I think we can make this world a better place. And there's already phase 1, 2, and 3 trials ongoing in diseases beyond the oncology field that hopefully will expand the ADC field beyond oncology. And so I really think that the future of ADCs looks bright. But it's important to remember what we've learned and where the future may take us. The future will take us to have more topo-1 ADCs in earlier indications, and we have a lot of opportunities because of this, but also challenges related to sequencing. To address these challenges, it will be important to innovate ADCs, and especially to the payload of the ADCs to limit cross-resistance with ADCs using sequence. And then, of course, we need better biomarkers, especially now that we're moving ADCs to the early setting, to the curative setting. We really want to understand which are the patients that need a HER2 ADC, a TROP2 ADC, a HER3 ADC, and there's a broad array of novel biomarkers that are being developed in the field, and I think will help to tailor treatment with ADCs in the future. And finally, we have to make ADCs safer. The idea from more than a century ago by Ehrlich of a magic bullet was a bullet that only targeted the tumor, did not hit normal tissues. And we're not yet there, but we're getting there. And I think it's important to make ADCs safer, more tolerable, and finally to use what we learn in oncology to expand their use outside of oncology. With this, I would like to thank Sara Toleni and Giuseppe Curigliano, my mentors, all the Dana-Farber colleagues and external collaborators that allowed the biomarker data that I've presented here, many friends that helped with development of this presentation, and funding support from Terry Broder, Sovereign Family, and Goldfarb, and all of you for your attention. Thank you very much. All right, and I think, does this work? Yes. I think at this point, we have still 20 minutes for Q&A and discussion, which I think will be precious given this amazing panel that we have. And I will kick it off with Professor Lambert. While we wait for people, please come at the microphones, ask questions if you have questions in mind, but I'll kick it off with a question. If you could choose only one part of an ADC to modify, which would be the most important for you based on your experience? The antibody, and so the target, the linker, or the payload?
I think that's an impossible question. Where I would say is that much of the advances have come in recent advances to go from the 2010s to today have been advances in actually the conjugation sites and the linkers to actually have stable conjugation so the linker payload doesn't fall off before the antibody actually gets to a target. And actually investigating the linker itself to actually tailor the cleavage properties so that the payload does come off in the target.
Thank you. This is extremely helpful. I see Professor Rim asking a question, please.
Yeah, thanks. Wonderful seminar. And this is a kind of a two-part question for anyone on the panel. The first question is, if you had a selector biomarker that could select between two possible say TROP2 and HER2 markers, would you use it? And second part, does it need to be proven prospectively before it gets tried?
So you know, this is a very interesting question because I have my personal idea on this. You have two different types of target antigen. Some of them can induce a clear oncogene addiction in the tumor. Some others are, let's say, stroma target or innocent bystander. So having both of them, I would select the one with a clear oncogene addiction, because up to now, it's quite clear that if you use an ADC against a specific target antigen that is a background for an oncogene addiction, you can observe a dramatic improvement in the outcome. And this is why maybe TDX is so effective in HER2-positive disease. So what is going to happen with other target antigen that are not related to an oncogene addiction? Maybe you need a higher expression in order to get more benefit. But in some disease, because it's also disease-oriented, since now we have also one agent approved in an agnostic context, TDX is approved for HER2-positive disease, whatever it is, but you know perfectly than me that HER2 is one thing in breast cancer, is another thing in gastric cancer, another one in colorectal cancer. So even if we like to be agnostic, we have to recognize that specific target antigen have a different biological role across disease. And this should be acknowledged when we are going to design clinical trial.
But can I just?
Sure.
Yeah. So I think I interpreted your question in a different way. And while I would agree with, yes, you'd go for the target that it is highly expressed or has an oncogenic addiction, I think we are in an era now where we can take advantage of going beyond just the target. It's an era where you can leverage artificial intelligence to get a composite of information about the disease pattern on that given patient, radiomics, quantitative computer scoring of the target, acknowledging the payload, the disease biology and whatnot, and really leverage this composite of information where, again, hopefully AI will help us make those decisions in a way that is smarter, that for a given patient, it will not be one size fits all. So we really need to personalize the treatment based on not just the expression of the target, but a composite of different things as well.
Thank you both. Microphone on the right.
Yes, first of all, thanks so much for this great session, this very nice overview of the ADCs. And my question is, can you, I guess at this point, speculate what would be like sequencing of the treatment versus concurrent treatment, like, for example, what is being done now with the EV plus SG. So what are the possible advantages versus disadvantages of doing like concurrent therapy versus the sequencing?
Giuseppe, what do you think? Should we combine ADCs or should we give them in sequence?
We have some clinical trials now addressing this specific point. Of course, you should work on the dose optimization. So potentially in order to combine together two different ADCs, you should reduce the actual dose. And to just cite what we just listened, I really believe that this concept of artificial intelligence that cannot be used in clinical practice can be really relevant if you have to combine ADC, because assuming you have two different target antigen, for sure, using digital pathology and artificial intelligence, you can better assess with the multiplexed analysis the concurrent expression of two different ADC, of two different target antigen, and you can better sequence, of course, the way you administer this. I mean, in theranostic, if you have something positive and you treat, and then it becomes negative, it means you can stop treatment. Maybe something similar can be developed also in the context of solid tumors, not with theranostic potentially, but with digital pathology, to better study the distribution of the target antigen and to better design the way to give a concurrent administration of the two ADC. So this is more future. I don't know how much of this can be translated in clinical practice, because you know perfectly that an ADC had a specific safety profile, and so this may affect potentially also the way in which we give concurrent administration of 2 ADCs. It's more a speculative question, personally, I believe.
So can I add something to what you said? So to me, and again, I'm going to take my corporate hat off, put my old clinician's hat on. I think it's all about the context of what you're trying to achieve. Because if we think about the way we treat breast cancer today, let's agree that the data up to this point suggests that in the metastatic setting, sequencing drugs is potentially the right thing to do, because at the end of the day, the overall survival does not seem to change if you combine drugs. But let's acknowledge that there are certain circumstances where you may need to do so to get a faster response, patients with visceral crisis or, using an example. But when you're talking about cure in the early setting, that's when we combine, let's use triple negative as an example, right? We are combining now anthracycline, Cytoxan, paclitaxel, carboplatin, pembrolizumab. That's 5 drugs in there. And it works, right? You have like a substantial proportion of patients now that are no longer recurring. So I think the notion of combining ADCs versus sequencing ADCs is going to be very contextual, depending on what the end point and the end game would be. And I go back to what I said before in my talk. It's all about the benefit-risk ratio. It's what are you willing to put up with in terms of added toxicity for a transformative outcome. And if the outcome is not transformative, it's not worth it. So I think juggling that, just like we do with chemotherapy, is ultimately a way to figure it out if we should combine or sequence ADCs.
Thank you. One question from the virtual audience I would like to ask Professor Lambert. How much of the activity of ADCs is related to an immune effect? And I will add, we've seen many trials showing wonderful PFS curves that plateau, just like immune checkpoint inhibitors do. Can we assume that part of that activity of TDM1, TDXE, and other agents is immune effect or not? What do you think about that?
I'm not sure I've seen any evidence really that an immune effect plays a big role in ADC. For a long time, I think I was an advocate of, for example, leaving IgG1, leaving the ADCC functions intact, but actually so that this effect could be manifest if it was there. But in truth, I haven't seen any data that suggests that that's helpful or hurtful. And in fact, it could contribute to, there's no data for it, but there's a lot of speculation that, for example, the ILD of TDSD could be exacerbated by the fact that the antibody component has intact ADCC, and there seems to be an inflammatory point there. Of course, I don't know it. No one's going to go back and make TDSD exactly as it is with the mutations to take out ADCC interaction. But I've come round in my thinking that for the data I've seen, most ADC activity is simply targeted chemotherapy. Now, it's not to say that because you're getting cell death in a more targeted way, so not the sort of blunderbuss of just cytotoxic chemotherapy that might take out all of the bone marrow and immune cells, that it might not be beneficial to have an ADC combined with, for example, checkpoint inhibitor in those diseases where the checkpoint inhibition actually is effective. And that certainly seems to be extremely effective in for example, in the Nectin-4 bladder cancer.
Thank you.
That's the prime example for that.
It's extremely interesting. And I guess this is also why many ADCs entering the clinic now are Fc receptor silenced to avoid ADCC, right?
Yes. I would say of the companies I consult with, half of the people are doing Fc silencing. It's a bit early to know now what difference this makes in the long run. One needs to study many more patients, but it doesn't seem to make much difference to the efficacy of the ADC.
Extremely interesting. So we have time for at least one more question. So microphone in the center.
Okay. Thank you. It's really a wonderful section. So I have a question. So comparing with the traditional chemotherapy drugs, what's the key limitations of the ADC drugs for the neurology disease and the solid tumors in the brain? Just following the audience questions.
So when you say neurology disease, you mean CNS metastasis?
Yes.
Okay. Giuseppe.
You know, we have a clear evidence, at least from some prospective trials, even if not randomized, that specifically trastuzumab deruxtecan work in brain mets. Just to remind to you, also in the DESTINY-Breast03, that was a prospective randomized trial comparing two ADC, there were some patients with baseline brain mets, and the activity of trastuzumab deruxtecan was very clear. I don't believe this is a challenge or a limitation. I believe this is a huge opportunity because in clinical practice, what we do usually with other ADC or with targeted agents is in case of brain-only progression, we stop treatment, we deliver radiation therapy, sometimes is a gamma knife, some other times is not a gamma knife, and then we start again with the treatment. Now, with the demonstration that TDX may give a response on brain mets, it means for me that if you have asymptomatic lesions, you can delay the use of radiotherapy, you can start and you can use trastuzumab deruxtecan, and if you achieve a response, you don't have to use radiation therapy. To be honest, this is what I am doing in clinical practice if I have small lesions and the patient is progressing to the previous line or eventually is ongoing with TDX. So I really believe this data, in my opinion, are practice changing. This will change potentially also the treatment algorithm because up to now, for active brain mets, there was a clear indication to tucatinib, trastuzumab, and capecitabine, since we have the evidence of a prospective randomized trial, but their two-climb was not designed in the setting of post-TDX. In their two-climb, patients received dual blockade and TDM1. So this is really an important information, and personally, I believe in a case-by-case discussion, some patients can for sure delay radiation therapy.
Thank you. Microphone on the right.
Hello, thank you for a wonderful session. It was really nice to hear. My name is Olav Engebråten from Oslo, Norway, and in Oslo we have done some research on internalization of ADCs. So could you comment upon the role of how internalization would actually influence the activity of the ADCs? We have some results that signals that is actually very important for the older Kadcyla type of ADCs and also for the newer ones. So I would very much like to hear your competent view on how this looks from your side.
Yes, I think internalization is an important factor. And so I'll answer your question in two ways, just for ways of thinking about it. First, HER2 itself. I mean, HER2 in normal cells, so what is probably HER2 low or 1+, actually is really a recycling receptor. It doesn't get degraded in the lysosome. It goes to the endosome and back out again. It stays in that space. And it's over only when it's massively overexpressed, as in HER2 high, that you get now some bleed through into the lysosome. I mean, it would be an equilibrium. That equilibrium level of, say, 1 million receptors on the surface is a function of making and degrading. And that's why TDM1 absolutely is only active in HER2 overexpression. Once you get to HER2 modest levels where the trafficking to lysosomes is actually fairly poor, it's not active at all. So, which also illustrates a second point. It depends on now your ADC design. So if you have a cleavable link that can be cleaved by proteases in the endosomes, actually extracellularly as well, you can still have activity, even if the internalization is poor. Now, another example that is quite well established in the ADC world is take c-Met as a target. It's often highly overexpressed. But ADCs have not been particularly active in c-Met expressing tumors unless that c-Met is due to gene amplification, which is where, if you like, the making of c-Met is just turned on all the time, and then you end up with high level of c-Met that is the equilibrium between making and degrading. But there's a lot of c-Met amplification, or where there's high expression of c-Met, but there's no gene amplification. And it appears that it's very poorly trafficked to lysosomes. And a lot of the c-Met ADCs are not particularly active. There is a company that I consult for that is developing as an ADC in the clinic right now, targeting c-Met, and it's a company, Mythic, and their technology is to engineer ADCs that actually let go of their target at the low pH of the endosome. So, for example, pH 5.5. And the contents of an endosome, if not attached to anything, end up in the lysosome. It's a sort of reverse of FcRn recycling, think of it that way. And the data is yet to emerge. There's been some early presentations, but that's sort of early analysis. But that's a long answer to your question, is that internalization and the route of internalization is important. So there are certain targets that may become effective if you can actually end up with more of the ADC in the lysosome if the target is a recycling target.
This fascinating, and I think it's a perfect way to close this session. Unfortunately, we are at time, but I think this also kind of brings us to how complex ADCs are. The target matters, internalization matters, the payload matters, but in the end, what matters the most, I think, is the benefit for the patients, and we're seeing that more and more. And so please, stay tuned for every trial of ADCs that is coming at this conference and beyond. And thank you all for attending this session, and to our wonderful panelists and speakers for joining this session. Thank you.